Implication of Hox and Meis PBX genes in acute leukemia was barely a curiosity 10 to 15 years ago, but it is now becoming a widespread phenomenon. For example, in human acute leukemia, whether acute myeloid leukemia, T-cell leukemia, or B-cell leukemia, we find that Hox genes are directly or indirectly involved in several of these leukemia, such that it is estimated currently that about 50% of human leukemias in general are induced by the misregulation of Hox genes and their cofactor. Interestingly, the two cofactors, Meis and PBX, are protein partners, and it appears that Meis is involved in regulating PBX nuclear trafficking, such that the cytoplasmic Meis PBX complex rapidly become nuclear, and one of the hypotheses is that together with Hox genes they target key genes involved in leukemogenesis. There are several observations which suggest that this model might not be right, and that in fact there's a Hox complex and a Meis PBX complex that might be involved in leukemogenesis. Our lab previously showed that HOXA9 collaborates with Meis1, but also with other fusion oncoproteins to accelerate leukemia development. HOXA9 does not collaborate with PBX to accelerate leukemia development. So for example, in a typical survival curve, HOXA9 will induce leukemia in mice in about 6 months, but HOXA9 plus Meis leukemia will develop in 1 to 2 months. We also showed that the phenotype of the acute myeloid leukemia which develops in an animal is Hox gene dependent. For example, HOXB3 induced leukemia, whether they are accelerated or not by Meis1, will have a very differentiated morphology, whereas the HOXA9 leukemia, whether they're accelerated or not by the co-expression of Meis, will have a very immature morphology. Our lab also worked in the generation of a transgenic mouse model for the E2A PBX pre-BALL. 2 lines of transgenic mice were developed, and these mice spontaneously developed mostly T-cell leukemia, not B-cell leukemia. Although, if one waits long enough, after 400 and some days, you start to see the appearance of some rare B-cell leukemias in both transgenic lines. In order to try to engineer pre-B leukemia in this E2A PBX transgenic model, we crossed the E2A PBX transgenic mice with CD3 epsilon knockout animals, which developed no mature T cells, and monitored leukemia development in the compound E2A PBX CD3 epsilon mutant animals. As predicted, the E2A PBX transgenic mice develop B-cell leukemia. Interestingly, the B-cell leukemia developed earlier than did the T-cell leukemia in the transgenic model, in which the mice were not backcrossed into the CD3 epsilon background. In order to get insights into the genes that might be involved in leukemia development in the E2A PBX transgenic CD3 epsilon, we infected the mice with the MMLV virus and found acceleration of B-cell leukemias as expected. We recovered the common insertion sites from transgenic animals and control tumors and found that the HOXA locus was targeted in 8 of the 9 tumors isolated from the transgenic mice and in 1 tumor from the control animal. Interestingly, the MMLV integration was found in a small region of the HOXA locus, as indicated here, located between the HOXA6 and the HOXA10 gene. Interestingly, the integration site in the HOXA locus, whether it was located between HOXA9, HOXA10 gene, HOXA3, HOXA10, etc., dramatically perturbed the expression level of several HOX genes, but HOXA7 was commonly deregulated between all of the leukemias that were generated from transgenic animals, but not from the control pre-B tumor, which also had an integration in the HOXA10 gene. The conclusions from this first part is that HOX genes and cofactors are important oncogenes in human leukemia, and our lab developed several mouse models to document this. PBX is not transforming unless flanked by a strong coactivator, such as found in the E2A PBX fusion protein. HOXA genes are activated in relevant mouse models of E2A PBX-induced leukemia. But the question from all of this is, how does this work? And from now on, our lab focused its efforts on understanding the molecular basis of Meis-induced acceleration of HOXA9 leukemia and of HOXA9 itself. 3 separate approaches were developed. First, we did a structure to function studies looking at Meis oncogenic activity and found that the PBX interaction motif, the homeodomain, and the C-terminal region are essential for its oncogenic potential. Interestingly, PREP1, which is a Meis-like gene, is not transforming, and in fact we see reduction in HOXA9 potency when cells are co-transfected with HOXA9 and PREP. Sequence alignment shows that the PBX interaction motif is well conserved between Meis and PREP, that the homeodomain is also well conserved, but this C-terminal region, which was shown recently to have transactivation potential, is not conserved. Again, we also showed that although Meis1 accelerates HOXA9-induced leukemia, PREP1 seems to postpone leukemia development. Studies from this first approach showed that HOX oncogenic collaboration is restricted to Meis, not PREP, that Meis, PBX interaction motif, and homeodomain are critical for leukemia acceleration, that the C-terminal domain of Meis is required for transformation, and that the C-terminal domain of Meis contains transactivation properties. And one big question that we've investigated is what interact with Meis and not with PREP. To more directly address this question, we generated a HOXA9 Meis-induced leukemia, isolated the leukemic cell population, generated a 2-hybrid library from these cells, and screened this leukemia using Meis as a bait. The positive clones were then counter-screened for interaction with PREP, and clones that specifically interact with Meis, but not PREP, were selected for further studies. The third approach that we've used is an approach developed in collaboration with Marc Terrien's laboratory. This is our fly-to-mouse approach. Importantly, the Meis PBX HOXA9 complex found in mammalian cells is also highly conserved in Drosophila. We thus decided to develop a fly leukemia model using the NUP98 HOXA9 human oncoprotein. Current studies indicate that the NUP98 HOXA9 induced phenotype in flies is dependent on cofactors which are known to be essential for HOX-induced leukemia development in mice. A large-scale EMS-based genetic screen was recently conducted, and several pathways that appear to be essential for NUP98 HOXA9 induced leukemia were identified.